vaccine. But next, I want to talk about really one of the top scientists in this country. I don't know if you know who she is, but I know you know where the story derives from. I think you remember there was a moment where science looked like it was going to do something spectacular, take a shift into a place, journey to a place we had never seen before. It was exciting, it was a ride, and it went and took a mysterious and interesting and somewhat devastating turn. Do you remember this in the news?
Stem cell research is about to take a giant step forward with the world's first human study using embryonic stem cells.
A cell that can grow into just about anything.
They're essentially unprogrammed or blank cells. Take a stem cell, manipulate it in the lab, and it can become a heart cell, a liver cell, a bone marrow cell. One source is aborted embryos. Another source, embryos infertile couples have set aside in a bank but don't end up using to start a pregnancy.
On the controversial edge tonight, stem cell research. It's a procedure polarizing America and the White House.
We don't believe that the average American wants to see tiny embryonic boys and girls, little children, used as experimental material.
It may be early to say that they are or aren't human life.
On the other side are stem cell researchers and celebrity patients who have lobbied Congress for federal funding.
I'm one of a million involuntary experts on Parkinson's disease.
Should federal tax dollars be used to destroy human life? My answer has to be no.
These boys and girls are not spare parts. It crosses a moral boundary that our decent society needs to respect, so I vetoed it.
So there it was. Yay, stem cells. Oh, aborted fetuses. We all watched this thing happen right before our eyes. It seemed like there was a miracle cure, and then we discovered that it came from aborted fetuses, and all of a sudden, this moral dilemma came up, and we had all of these issues, and then finally, George Bush pissing off the entire scientific movement, saying, I am not going to use babies and embryos as machinery or to make drugs or any kind of medicine. This is not moral and it's not allowed. And there was this real, I feel like, dark cloud came over science and came over this nation as we thought, did we just pass up great science? We just pass up an ability to heal ourselves, maybe even without drugs? But what I didn't know was there's an entire different story that was happening at the exact same time. And just like every other scientific story we now look at, there was another idea. This was not the death of stem cells. It was death of a story from the pharmaceutical industry and our government agencies that wanted to use aborted fetal stem cells. But did you know that somebody else had an even better idea at the exact same time? Take a look. This is Dr. Theresa Deisher.
Hello, I'm Dr. Theresa Deisher with Sound Choice Pharmaceutical Institute, a nonprofit research organization whose mission is to conduct scientific research that will lead to the replacement of drugs and medicines that are discovered through the exploitation of another human being. Embryonic stem cells are so primitive, they grow out of control and they form tumors 100% of the time. I was the first person to identify and actually patent stem cells that I isolated from adults. It doesn't form tumors. So the controversy surrounding my discovery was that it was not necessary to destroy an embryo and able to do the type of work that people wanted to do. There were many scientists absolutely outraged over my discovery. There was a battle because the embryonic stem cell proponents had won the public and the government, and they actively blocked the advancement of adult stem cells. There is no reason that human beings have to be exploited in biomedical research, and we are leading the way to alternatives. The opinion of the experts in the field was that stem cells did not exist in the adult body outside of the blood system. And that was why people wanted to work with embryonic stem cells or fetal stem cells. And I had done some work with adult blood-forming stem cells And in an experiment where I was working with adult mouse hearts, we stumbled upon it, an interesting stem cell in the culture that was not a blood stem cell. And so I followed it. I knew what a stem cell looked like and how it acted. And it was just incredibly primitive. And it could become almost any kind of cell in the body. And we called it, one of my colleagues called it the ultimate stem cell. People who have since rediscovered it, it's been repeated, call it a V cell.
A V cell.
A very small embryonic-like cell. I called it a pluripotent mesoderm-derived cell.
I won't even attempt to restate that myself. So when you made this discovery, did the medical community just say, oh, thank God, Dr. Theresa Deisher has an answer. We can get out of this moral issue with aborted fetuses and move into this cell which does everything that we're dreaming a stem cell could do? Were you celebrated for that?
Well, I, at the time, was lucky to be collaborating with some of the biggest names in the cardiovascular field in the world. And I brought in some of my collaborators to see this cell. I remember Mike Schneider, who's now the dean of the cardiology department, I think in Royal College in London. Maybe I'm wrong, I'll have to look that up. But he was at Baylor then, and really a top name in cardiac disease, and came in and he looked at the cells and I described them and he said, oh my God, I could kick myself. I've been throwing those away for decades. I thought it was debris. They don't realize how small stem cells are. And then his group went back and they used a similar methodology and pulled out stem cells. They called them side population cells. And other cardiac people who I brought in, I mean, were overwhelmed. They couldn't believe the types of cells that they had differentiated into. And I think it kind of helped trigger independent research and discoveries in that field, because when you go out to repeat it, you don't always do exactly the same thing. And so they would identify a slightly different stem cell, but still an adult stem cell, which really built the field. But for people who wanted to do embryonic stem cell research, I was for a very irrational reason, a very hated figure. And that's because when pluripotent cells exist in the adult, there's no need to exploit other human beings to do that research or to develop cures for patients. And some of those programs were actually shut down because of my discovery. And they were very angry about that. I didn't try to shut them down. The other scientists in the company did, because there's no reason to compromise your morals anymore. And I would just say I was shocked by the irrational behavior, the hostility, and the attacks, and not very scientific.
When I heard about that, I was, I always question like the motives of why was all the media about, aborted fetuses and stem cells, and I don't remember really hearing a lot about these adult stem cells, at least not till more recent. Now on the doctors, we're extracting blood and doing vampire facials, where they're injecting, your own stem cells back into your face, which I hope you're getting kickback on every vampire facial the Kardashians are doing.
Get a penny from it. But that's why there was so much resistance to the adult stem cells. Embryonic stem cells were patented. And in academics, when you have a patent, if it becomes commercialized and any payments along the way, the scientist gets 1%, 2%, 4%, up to 8%.
Right.
It's a lot of money in their pockets. And so scientifically, they're going to promote that. Whereas working in a company, we don't get anything from our patents. Actually, I got $1 for every patent. My interests were in the health of the patient and what's best for the patient. And nobody can really own your own stem cells.
Right.
Right?
Right.
And so because of that desire to make money, right, and because of a lack of patents in the adult stem cell field, there was a tendency to go for embryonic. And what the government should have done, but they were horribly misled, was step in and fund the adult stem cell research. A company isn't going to fund adult stem cell research because they can't patent.
Well, you know, this brings up because we were looking at this Alfie story at the top of the show. And this, you know, and this is something that the more I look into this issue, these things are much more complicated than we tend to think about. You know, we think about pharma as just a bad guy, or, you know, but there is, or that government shouldn't be involved in medicine at all. Or I will often say things like, I don't think that the government should be investing or giving money to anything. Just take it all out and let the market decide. But the problem with that's sort of a libertarian thought. This is why, again, folks, I always say I can't find a home to rest now politically, because every issue, you know, every one of these parties or ideas seems to have a flaw. And what you're pointing out to me is a flaw in what I think is a libertarian stance, which is, you know, let the market decide. But if the market decides, then it's financing that's going to decide it. And just like you're saying, if the finance of something is the only way it gets studied and it has to show profit, then we get stuck in this place where you have to have patents. And if you have to have patents, we're not going to look at something you can't patent, even though it's the best idea there is. So you're actually saying there is a place for government to step in and say, you should be funding research in something that is not as highly regarded or wanted or needed or is financially viable as the things, the most important or the most powerful industry you work for, the pharmaceutical industry. They're not interested. So, you know, how should that balance work?
Well, I mean, the mandate of the NIH and our government is the best interests of the public and the public's health. And so in that case, where, I mean, adult stem cells are always going to be best. Your own stem cells, whenever possible, they're always going to be best. That's a no-brainer. And that needs government funding. That's their mandate, where the commercial sector won't move it forward because there are financial returns for them. When it's the case that there's huge financial returns in the commercial sector, then I would say the government doesn't have a place there. Or, I mean, we can go back to vaccines, right?
Yeah.
You know, the mandatory aspect of our vaccine program, it's basically mandatory, creates billions of dollars for these companies, yet they have no liability. That's the strangest situation you can possibly imagine, and certainly not a situation where we're going to have safety as a consideration.
So when we talk about the considerations of government, there was actually a shift in this story. So we elected a new president after George Bush had shut down stem cells. Then all of a sudden came Barack Obama. Do you remember this moment in the news?
Today, with the executive order I am about to sign, we will bring the change that so many scientists and researchers, doctors and innovators, patients and loved ones have hoped for and fought for these past 8 years. We will lift the ban on federal funding for promising embryonic stem cell research.
Now, you couldn't hear that, but that was Barack Obama, you know, essentially saying we're lifting the ban on the use of embryonic stem cells. What were you thinking when you watched that happening in the news?
What a waste of money. What an absolute waste of money. I had been involved in the embryonic stem cell debate for several years, and that was because of my experience with adult stem cells. I had always been scientifically opposed to embryonic stem cells, and at that time in my career, I would actually have to say I was quite the atheist. Mm-hmm. But I was opposed from a scientific reason, because embryonic stem cells are by nature tumor forming. And that's how we define them. We put them in models, and if they don't develop tumors, they're not embryonic stem cells.
Wow.
Why would you want to give someone a tumor forming cell? That's crazy. And in every animal model for decades, those cells had formed tumors. We had never been able to overcome it without losing the stem cell characteristics of the cell. And then another aspect that I was always against, that is just an incredibly expensive therapeutic. You're talking hundreds of thousands of dollars, $500,000. When we could use a patient's own stem cells, and in many cases you would be talking about $5,000 to maybe $25,000, depending on the complications of their disease. That's a crazy waste of public money, and embryonic stem cells are patented. If they're that great, you know, the biotech and pharma will be developing them. But the truth was,
right.
the biotech and pharma weren't developing him because it was a stupid idea.
And you didn't just sit there when you watched that news. You actually took the bull by the horns and you sued. Who did you sue?
We sued President Obama and Kathleen Sebelius at the time, who was the Secretary of Health and Human Services.
Sounds familiar, right? People that do things like that.
I did not initiate the lawsuit, but I agreed to take part in it, not only to allow my name, but to be scientifically active in the lawsuit. And that was because what President Obama did was he diverted funds from adult stem cells into embryonic stem cells, which was a complete betrayal of the American public. And so we sued to stop that. We won, we lost, we won, we lost. Ultimately, we lost at the Supreme Court. But during the time that that lawsuit was active, what it did was it allowed scientists who wanted to work on adult stem cells to come out of the closet without fear of losing their careers. And when a scientist would come out against embryonic,
what is it with science, Theresa? And I saw this on the Doctor's Television show. I've said it before. When I would find a surgeon doing something so like I remember one guy that did a brain surgery and I was scrubbing in. I was like getting ready with my camera, and this other surgeon said to me, "Who are you videotaping today?" And I said, "Who?" And he said, "You're about to watch a Jedi at work." This guy is doing things that amazes all of us. And that guy was under so much for his license, constantly other doctors suing him to take his license. What is it about science? I was saying to Jimmy on my team the other day that adult stem cells at the same time you had, fetal stem cells. Why does it seem throughout science, we always, when we have a diversion in the road, we always take the wrong road? Why is it the bad idea is always the one, and that anyone that stands up for the truth gets attacked? What is it about science that attacks the people with the best ideas?
I don't know. I guess I can say this because I'm a scientist, but you can't.
Right.
I think that there is, in many cases, an arrogance among MDs and PhDs that they don't want to admit that they're wrong. They don't want to question. Sometimes they'll hold to dogma where there's absolutely no data anywhere that supports it. But these are also very confident people, and you have to be confident to be a doctor or scientist, right? Mm-hmm. And so, yet that strength, if it's not balanced with perspective and curiosity and inquisitiveness, is a huge weakness.
Right.
So confidence can then become arrogance. And arrogance does not like to be wrong. And if that arrogance is based on a really weak foundation, right?
Right.
Then it's attack because they're arrogant. Really, they're not confident. That confidence,
there's a difference.
They lost the balance, and so it's, turned into arrogance. And arrogance is weak, actually, and vulnerable, and they're very angry when they feel that they're attacked.
I think you see that now when I look at this, these statements scientists make about anti-vaxxers, the rage and the anger, when we keep putting up more and more science being done around the world, showing that vaccines are not as safe and not as effective as we had once thought. I want to get into some of the work that you're doing there. So your work in stem cells led you into looking at, and also I think your issue with, fetal tissue and fetal, stem cells. We know that the MMR vaccine has aborted fetal tissue in it. We know that the chickenpox vaccine has aborted fetal tissue in it. And for many, that's just a religious issue. It just doesn't seem right that I should be injecting the DNA of a baby, of a dead baby, into my healthy living baby. And it seems like an abomination when we think of it from a religious perspective. But from a scientific perspective, you actually brought something to this discussion I never thought about. And I find it interesting because I worked with Andy Wakefield, and Andy Wakefield was all about the MMR vaccine. Yet at the same time, the real discussion that sort of stole the day was thimerosal and mercury. That mercury what was dangerous in vaccines, and that the MMR was safe. That the MMR never had aluminum, never had mercury in it because it's a live virus vaccine. And so, as being new to this and coming from the doctors as a medical journalist, all I said, and I kept thinking to myself as I'm doing this stuff, the movie was I have a whistleblower at the CDC saying that they did a study on MMR and it appeared to them that the vaccine was causing autism, at least an increased risk of autism. Andy Wakefield came to that similar, conclusion. But I was thinking, without mercury or aluminum, though, what is it that would be causing that? And you actually have been working on a scientific explanation for that. Can you tell me what that is about?
Correct. So the vaccines don't have human fetal tissue in them. They have debris from the human fetal cell line that was used to manufacture them.
Okay.
I have been in commercial biotechnology, Genentech, Immunex, Amgen, biggest names in the world, for a long time, 1990, actually, 1988. And I worked from basic discovery research and clinical development. So I've been exposed to insertional mutagenesis and small fragment homologous recombination and the dangers of
okay, layman's terms, really quick, because those are really big words. So insertional, okay.
So when you make a vaccine or when you make a biologic drug, it's really expensive to make them in a test tube. And so we mimic nature's way to do that, and we make them in a cell. And then we try to purify the drug away from the cell. But that purification process is never 100%. And that's because your yield would be so low, you wouldn't be able to sell your drug. So there's leftover DNA from the cell line and cellular debris. So when we use a hamster cell, there's some hamster DNA. But a hamster is a hamster. It's not human. And we recognize it as foreign and eliminate it.
Mm-hmm. Our body does. Our body recognizes that this is not human DNA.
It's not human DNA, and it's not decorated like a human decorates their DNA. So DNA is not just ATCG, it's decorated. And the decoration is absolutely species specific. Okay? Humans decorate their DNA one way, hamsters decorate them another way, and the two never meet, right?
Yep.
So when we go to using a human cell, we have human DNA in our final product. And that human DNA is human and has the potential, it's well documented science. If I were to bring in all the papers, there'd be stacks this high, hundreds and hundreds of papers from gene therapy and biology and immunology establishing this. It can cause autoimmunity, and it can be readily taken up by adult stem cells. For whatever reason, they readily take up DNA fragments and insert into the genome and cause mutations.
So your own DNA mutates. You sort of absorb these letters because they're decorated like human beings would, and our stem cell goes boom and changes its own code, essentially. Is that correct?
Yes. And you can have a mutation in one stem cell that can affect 60, 70, 85% of the blood cells in your body, because the way our blood system is set up, there are 8 stem cells that replicate themselves and then make all of the blood cells in our body. And usually one is dominant. So a mutation in a stem cell could lead to, general autoimmune disease, general dysfunction, because that stem cell makes the cells in your body that regulate immunity, including in your brain.
Wow.
So we have been investigating that. And when you look at autism, there are distinct years where autism will suddenly jump, the curve of its rise. It's called a change point. And when we look at change points in different countries all over the world, they are always, so far, associated with the introduction of a human fetal manufactured vaccine. Now,
Wow.
I think that the biology is very
So meaning
complex.
at those points, you can see it inserted into, a country.
Yes.
And you watch it jump now in their autism rate
Goes up.
upon that delivery.
Yes. And the more children who are vaccinated with it, it continues, the prevalence continues to go up. So the disease, all diseases are complex, and I believe thimerosal can contribute and aluminum can contribute. And I believe that Dr. Wakefield's concern about the measles virus being retained in the gut can certainly contribute and is part of this complex biology and pathology that we're seeing in children.
Mm-hmm.
But we're really focused on the fetal contaminants because it is the only vaccine change that is universally and temporarily associated with these change points.
Wow. And so your study, you just did a study this last year. Can you tell me a little bit about that? Like how, what you were looking at and how did it turn out? What did you see?
Absolutely. So scientists trying to do gene therapy have learned that big genes don't get into the genome very well. And so they chop up their genes. They do little fragments. That's why it's called small fragment homologous recombination.
Okay.
And they have demonstrated that stem cells readily take up these small fragments.
Because we wouldn't have, I guess one of the issues that, when you talk to big scientists in the field, like maybe Paul Offit or Stanley Plotkin, I think the assumption is, well, yeah, there's fragments of DNA, but even if it affected one of your billions of cells or whatever, what difference does it make? But it would only be a problem if it's not just any cell, but our stem cells, correct?
And it's the stem cells that take them up. So, and they've treated animals, right? And the stem cells readily take them up. And these are scientists trying to do this for gene therapy.
Right, to go in and we want to insert a
and
fix
correct
into a problem,
a gene.
correct a gene. We'd love for it to deliver right where we want it to deliver in the stem cells, but
it does.
It does.
So stem cells readily take this up. And so what we did was we actually were just trying to continue to close the loop. So we took human stem cells, and in the body, adult blood stem cells grow really well. But for whatever reason, when we take them out, they don't grow. So what we did was we took adult stem cells out of the body, we ordered them, and we put them in culture where they don't grow, and we added vaccines to them. And we added the fetal manufactured vaccines and the DTaP. And what we were looking for was survival. So if the cells survive, it means a mutation event has occurred.
It should have died.
Because they should have died.
Okay.
And they did survive. So they survived. We took it out for about 3 months and grew and whatnot. And we want to go back based on what we learned from this first study. We want to replicate those findings. And we want to design a study so that we can continue to grow the cells and then actually inject them into animals and ultimately look for tumorigenesis or other biological.
Now you didn't just have, you had several groups, right? I was looking at this, but I didn't fully understand. You had something to do with the chemotherapy, wasn't there?
So
tell me about that.
we know that chemotherapy in low doses causes mutations. Okay. So what we did was we used this chemotherapeutic in low doses. That was our positive control.
Okay.
Would it induce mutations, like said in the literature, and induce survival? And it did. So it increased survival by 2-fold.
So the normal cells should die.
Yes.
With the chemotherapy, it increased 2-fold, like it proliferated on some level.
Yes.
Okay. And so that shows you that, so you're looking at normal cells. Now let's see when we give them chemotherapy, and that does what we thought. It's mutating and proliferating in a way we don't want them to.
Right.
And then you looked at the group that got the vaccines. And how did they respond? Did they respond similarly to the chemotherapy?
No, it was actually a much greater response, about a 5-fold response.
So two
times more than the chemotherapy we'd call our positive control.
Right. Yeah. Wow, fascinating. So it could possibly be even more dangerous on a very simple term than chemotherapy itself, that injecting these fragmented stem cells could be causing mutations that, and what does that lead to then? In the end, what are you looking? Is it just autism? Or are we talking about just autoimmune disease? Or do you think cancer is involved? Like, what do you?
Well, I don't think that every childhood disease is involved in this aspect of vaccines. I want to be very clear. People want to take this and blame it on everything. I don't. I clearly think this is involved in autism. And in 2010, I spoke to the Washington State Medical Association. And I had actually expected to be humored on my pro-life views and attacked on my science. And the opposite happened. After I spoke, a pediatric oncologist spoke and strongly objected to my use of the word electively aborted fetal cells. So she said, just, you can call them human fetal cells. However, she had been involved in humanitarian vaccination campaigns with the MMR and in Mexico. And after those campaigns, those children started getting leukemias and lymphomas that were unheard of in those areas before. And a King County executive actually spoke and raised concerns about the MMR and vaccination campaigns. So I did a little research, and there is indeed elevations in childhood cancers. Wow. And the data is not quite as robust as with the autism. So it was in May of 2014, actually, that I finally said to my group, okay, now we're going to turn and we're going to put this intense effort to really dig out the hard numbers and do what we've done with autism in every country. We're going to do this with childhood cancers. And we began to do that. And I guess it's ironic that just a few months later, my own son was diagnosed with what was once an unheard of childhood and adolescent lymphoma called Burkitt's lymphoma. And I can tell you firsthand from being on the cancer ward with my son, when 3 of his friends turned up, and I'm not kidding you, with cancers. And I mean, they can't build these childhood cancer wards fast enough. And I believe that the cancer increase is very strongly linked to the chickenpox, which is so heavily contaminated. There is more contaminating DNA in that vaccine than viral DNA.
Wow. All right. So obviously, you know, when you do stem cell research, there's bio firms and there's money available there. When you are getting into vaccines, is it easy to get funding? I mean, when you're taking, I mean, you have, and I asked you this, you know, when we were on a phone call a few weeks ago, you know, how has this been for your career to jump into this world of vaccines? And you said, you know, if I was doing stem cell research, I could probably get millions of dollars for that study tomorrow. But when it comes to vaccines, I have to look somewhere else.
It's very difficult. We've applied for government grants. Repeatedly, we're turned down regularly for absolutely absurd reasons. I didn't want to get involved in the vaccine and autism controversy at all. I have worked on controversial topics since I was a graduate student. I don't really choose them. They just seem to be controversial. But I think I mentioned I worked on beta blockers for heart failure as a graduate student, which was very controversial because the key opinion leaders, the experts, just decided that beta blockers were contraindicated in heart failure. And it was extremely controversial. And then I got involved in the stem cell field and the embryonic. But when I was asked to manufacture alternative vaccines to the human fetal manufactured vaccines, and I reluctantly said yes. It wasn't my area. Stem cells were my area. And I began reading about the subject. I mean, I couldn't miss the vaccine autism controversy. I hadn't been familiar with that before. And as I read about that, and I read the scientific publications that supposedly discredited all of Dr. Wakefield's work, and you looked at the data, not at what they said, the data actually very clearly supported that there was a change point around 1988, and right when he was doing this work. And what really hit me was the only thing that had changed So that's when they changed the manufacturing from animal to fetal. And we took a quick look around the world, and it seemed to hold up, and did quite a lot of discussion and debate about this, that it would hurt our work, our ability to raise money. But it was just unconscionable not to do the research because the connection, it is so overwhelming.
All right, folks. So today I told you we're celebrating great scientists, scientists that follow where the science is going. I think in this discussion, you can clearly see that's the case of what Dr. Theresa Deisher is doing. But you can get involved right now. You know, obviously our government is not doing a very good job of investigating vaccines, of safety. They want to say the science is settled. Clearly, you know, this is just one of many different discussions that prove that the science is not settled. But you can make a difference here today. Dr. Deisher is doing something that you never hear about. And honestly, repeating a study. She had success on her study last year showing that these vaccines are, in fact, affecting our stem cells. She wants to take it another step. Instead of watching the news like you always do, we say here at Highwire, we are the news. We are not watching the news anymore. We're not reporting the news. We're making the news. We are here to make a change. You are a part of Highwire, and today I want you to do something. Right now, you should be able to see where you can go to. You can always type "I can" into your comment box, and we will get you the information. But I want you, everyone out there right now watching, can at least donate $5 to this study this year so that we can take it another step. If you have more, please give more, because we are the only way this is going to change. If you are waiting for pharma, the most powerful lobby in Washington, to do what is right as they fight for patents and to make more and more money, you're wrong. You can make a difference. We are making a difference. Today, go to sleep tonight and say, you know what? I made a difference. I helped a real scientist that backed her way into this vaccine discussion to do something important, to really go further into investigation, to back up what she's already seen in one study. If she can show it again, if it happens again, if we take this another level, do you realize what that is going to do, where this discussion is going to have to go? You can make a difference. Please donate to, and you have a nonprofit, so it's a nonprofit donation. Please go and donate to this cause, because that is how we make change. It is time for us to get involved, and today you can do something spectacular. Dr. Deisher, it is an absolute honor and a pleasure to have got to spend this time with you. For those of you out there, I know this is a longer, when on the doctors, they always say 5-7 minutes is the longest you better go with a discussion. But I'm sorry, folks. I want the details, and it was really, really informative to get to talk to you. We're here. We're going to continue to shine a light on your work, and hopefully you raise the funds so that you can continue to move forward in your investigation. I just really want to thank you for taking the time and joining us, and look at the beautiful weather we got here.
Welcome to beautiful Seattle. Thank you for having me on.
Keep up the great work.
Thank you.
All right, thank you.